work. It's just like looking at the TM, which is in PCR there is a specific temperature at which something anneals, and that differs depending on what species it is.
Yeah, I think there is another question from Ponraj. He's asking that he's not able to understand about microsatellites. Can you please explain again?
Yeah, microsatellites again are one of those techniques that are kind of, it's on its way out. So this is based on repeats of specific parts of the genome. So some repeat, some are let's say dinucleotides or tri, and they repeat for certain numbers of times. So so it goes again, like, so if you can have a dinucleotide which is 80, and then this can repeat 10 times. So which means if you sequence it, it's 80, 80, 80, 80, 80, like into 10, 80 into 10. Yeah. And you can have trinucleotides, which is like, TAG or something like that. And then it can go like TAG 10 times. Now, the whole idea is that because there is a lot of variation, every individual has a slightly different number of repeats. So if you target a specific microsatellite, which means it's a specific, if this TAG, you can look at how many times it repeats. And with that, you can use it for looking at differences. So you can use it for looking at individual differences for like DNA fingerprinting. That was what was used even in crime and things like that. Yeah. So I think that's what microsats are. Again, this is a technology from when you did not need to sequence. You're only looking at the number of repeats. You don't actually sequence you don't get the sequence data for looking at this. Now, mostly people do things slightly differently.
There is another question from discussion forum again from Prishandi. So she wants to know what information can a hybridized DNA give? How does that play into the taxonomical differentiation?
Yeah, so this is like I was saying, this is almost like uh, history lesson, the DNA hybridization. It's like you look at, ancient guns and say, oh, wow, the people used to use this in warfare and it used to work. Um, so, um, uh, so yeah, it's not incorrect. It's just that it's not very specific. So based on, essentially the these work with pairs of sequences. Let me hold on. This is a longer discussion. Just give me a moment. Sorry, I'm trying to find a good reference that you can use to kind of understand this, because obviously it's not going to be a very yeah, I found something, and you can try this. Yeah, I mean, so what is needed to understand in this is that when you're making phylogenetic trees, it's based on some numbers. It's a similarity about how similar or different two species are, and how you get that number is what differs. So you could do it earlier by looking at traits. Yeah, and more recently, sorry, I got a little yeah. So earlier it could be done just by traits, and then later by using genetic data, you could look at number of differences. Yeah, I think what these people were doing was looking at the differences in temperature. So you can look at that link that I have shared. That gives a very rough idea of what's happening.
Robin, have you shared a link in the chat box?
Yeah, I've shared one link which is written and another which is a slide share. I can't vouch for the accuracy of what is in the slide share, this thing, but I think
Because
it.
I really couldn't find it in the chat box. Has anyone found this thing link? Okay, now I think
No, I
now
think it went to it was going to only the IIT, to the
okay,
host.
okay. Now it's visible like this. Yeah.
My bad.
Yeah.
I'll send the other link as well. Yeah, so you can read about this. There's slide 4 on that slide share that kind of gives you some idea.
Yeah. Robin, there is another question again from Prashanti. So he's asking, exons are translatable parts. Doesn't that mean that they are expressive genes, hence useful than the introns? Without prior knowledge of complete genome sequence of the species, how to choose which part to take?
Yeah. I don't know, Prasanthi. The thing is that you're asking a lot of. See, I'll tell you. First thing is that the motivation of this one lecture was to just provide a brief overview of what are the techniques that are used. And I think we already went very far in along that line. But what I can do is provide some general information. And yeah, so the idea of, in general, what you're trying to do with molecular techniques, there are so many different things, right? So we are uh, 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 the thing about trying to use or trying to talk about a topic like molecular techniques is that it is really wide, because people use it for different reasons. Yeah. And I think that mostly what we're trying to do when we're doing phylogenetics is you're looking for things that are not under selection. Which is when you're looking at studies which are, you want to look at neutral regions of the genome. So that this is what gives you an idea of relationships between species. You're not looking at selection, but there are studies that also look at that. That depends on the kinds of studies that people do. So exons, as you said, they are kind of highly conserved. So depending on how much variation you want and what is your question, you use different genes. I think that is the best way to kind of put this. So today, there's at least in bird phylogenetics, ultra conserved elements are very common. Just excuse me. Sorry. Yeah, I'm a bit down with a bad cold. Yeah. So what these ultra conserved elements are they are conserved bits of the genome which are conserved through tetrapods. So all tetrapods have this. And now what these really smart guys did was they developed these probes which are targeted at those highly conserved elements. So this is the ultra conserved elements. And then they use this in next generation sequencing technique. So you get basically the flanking regions around that conserved elements. One second, I'll find you a link for that. Oh. Yeah, I think you can go to this website, and that gives you an idea of how people have been using UCEs, these ultra conserved elements. Yeah, and now there are these UCEs identified for different taxonomic groups as well. Actually, there's a very nice presentation which I'm looking for. Just hold on. So if you scroll down on that website, you'll see all of the resources that are there for that, but that's not what. Yeah, you can look at this FAQ. That can the first question is quite useful. There's a very nice talk and a PowerPoint called Phylogenetic Nirvana, and that is what actually I'm looking for, which actually answers that question that was asked. Yeah, sorry, I'm missing a few other questions now. Sachin has asked a question: do genes control or play any role in traits like parenting or sexual selection? I mean, that's a bit tricky to say that that is how it works. I mean, this is the golden you know thing that researchers are looking for, and how these traits evolve. But mostly what people find are relationships. Whether they are causal or not is a bit more difficult, because there is a big interaction that happens among various things, various ecological things, like, for example, resource. So there are some species, for example, that have biparental care, and they have they can facultatively switch also. They can have both parents, they can have male only, they can have female only, and they can also switch. So it can depend on various things. So these are difficult things. People do study in one of the talks. I think I talked about uh, these relationships of different species on a phylogenetic tree, and then you can look at traits on that tree, and you can try and see if there are some conserved um, uh, evolutionary traits. That is possible, and you may be able to recover that. But I don't think finding specific genes, you can find some associations, but whether they are the true causal thing is difficult to tell. There's one more question about subspecies. Yeah, I think the entire idea of what is a species and subspecies is a human construct. It is made by humans to help us classify life and diversity. So, like most human constructs, we'll have to keep changing it. And in this case, we change it based on new information that comes up. So as we learn more, we have to modify our definitions of uh, these constructs that we have created for our own ease. Sometimes it complicates things. So the idea of subspecies is exactly in that domain. Initial ideas were that people said species are these true biological species where there is no interbreeding, and subspecies are, close to that, but they may interbreed. But now I think people know that there are models of speciation with gene flow, And hybridization is rampant. So it's a little bit more challenging to draw this line. It's like drawing a line in the sand and expecting species to kind of stick to that. So there is, so having completely muddled that idea now in your heads, I can say that at every point of time, people doing research on species, subspecies, and all that, everyone has a fair bit of understanding of what is currently accepted. And I can tell you what is currently accepted is that you need multiple lines of evidence. So this is where we are at currently. So you can't just, especially with the bird work, it's very clear that you can't just have genetic data. You need acoustic data. You need some other ecological data. You may want to add morphology. All of these taken together, if they are different, that is what I think goes to identifying a species. How different is a little more debatable. And ornithology, the taxonomy ornithology is a bit conservative, and it's very tightly guarded by how do you say this? By some sections of the society, let's say. Yeah, that makes sense. Yeah. Sorry for the very long answer, Govind.
If anyone has questions, I think you can ask directly also. That wouldn't be a problem because we don't have too
Sure.
many. Yeah.
So it's a generic question. Is there a central repository of all these DNA sequencing, this data, or the basically the specimen maybe or samples basically, which is controlled by some governing body or something?
Yeah, there's no real governing body as such, but there are various repositories. One of the most common ones I've just shared that. This is where a lot of data goes, and you can go, if you're on that page now, you can see if you first toggle that first thing at the top, and you can scroll down to, I think, yeah, there's like a lot of different things you can. I'm just trying to think of what would give you. Yeah, I think you can just select maybe just select gene, and sorry, I'm just trying to do this and then tell you so that you can
I just entered Babbler there in the gene section and it gave me some list of data related to that.
Yeah, you got that right. Yeah, so basically that's it. So and you'll see some data from our lab and things like that. So it's fairly what you should do is look at this thing called Popset. Those are nice ones to explore. So if you do Popset and let's say you click on lion or something like that, I think that'll that'll give you some yeah.
So Popset is related to a generic population, you're saying?
No. See, what I was trying to do was make things a little bit easier for you. So Popset are sets of genes that are used in specific studies. So you can view the entire. For example, I study these birds called shortwings in the Western Ghats, and you find some in the north of Palghat Gap, you find some in the south of Palghat Gap. And we use different genes like cytochrome B, ND2, many different genes. So if you have to actually try and find all of the information about these shortwings or Sholicula as they're known today, that is a bit challenging because you have to enter Sholicula, look for which genes, and then download them and then use them. But Popset actually puts all of them together. So they are just easy set of genes that you can play around with. So yeah, it's just a simpler thing, and they have some visualizations also, which is pretty cool.
This is accessible to anybody?
Yeah. It's a public, it's a literally a public repository. Anyone can use it. Anyone can access it. All the data, all our data from Western Ghat birds, everything is there. You can, just look through it and you'll find all of the data.
On the first web page, I saw that there was analyze and all these tabs available. So does it mean that we need to, we just submit a sample and then gets done easily there?
No.
Or is there a subscription based on that?
Yeah, no, actually they have everything is free on NCBI. It's not charged. But when you say analyze, what they will just do is they'll just it's just a bioinformatic analysis of the sequences that are there. So it'll tell you how much similarity one has to another and things like that. But to be very honest, there are a lot of things in NCBI, and I'm not sure if I know everything. Yeah.
Just one more question. Expense-wise, okay, or cost-wise, I remember that there was a decision on that. I think either it was a dodo or like a pink editor. They did some analysis using the spectrometry, if I'm not wrong. So, how does it compare with the DNA sequencing or genomic related study? Which one is preferred and when? Are they one cost effective over the other, something like that?
Yeah, this is Sachin, I just need a little bit of more information of you to help answer this question. What do you do?
I am an engineer, software engineer.
Okay.
And but this is a curiosity question. Like
no worries. I mean, like basically, if it was an undergrad, doing somewhere, I would answer that slightly differently.
Okay.
So
whatever way I mean.
Yeah. No. So I'll tell you. So I'll give you examples from the bird world because we are in basic ornithology. So in the bird world today, the baseline is just a few genes. So you need some gene sequences with a cytochrome B and ND2. These are the most popular ones that people have used. And in fact, a lot of people will go by this thumb rule of how different species are. Just look at the cytochrome B sequence differences. And if it is about 2%, and there's this thing called clock, which I think I explained in one of the this thing. So every 2% divergence of cytochrome B roughly translates to a million years or so of divergence. So it's so people use like these thumb rules, but they quickly fall apart because there are lots of there are a lot of exceptions. And today, mitochondrial DNA alone is no longer acceptable because there are so many exceptions. So you have to combine mitochondrial with some nuclear DNA. And then came the question of how many genes are enough to tell a story, which means that, basically everybody is going for the truth. What is the truth of how different two species are? Or you think they're different, so you're going to try and do variety of data you're going to add. And you can say that the ultimate is to get the entire genome, but that is just ridiculously expensive, and you don't need to do it for just answering a very simple question. So people, depending on the complexity of the question, keep adding more and more genes. And we went from just having, I don't know, in 1970s, we didn't have sequences, as I was saying in the the lecture. And then we went to DNA sequences. One or two sequences would be enough. Then people started saying how many base pairs, how long those sequences are, how independent those sequences are. Do they tell a different story? So, yeah. So uh, uh, it's a difficult question to answer, in one simple this thing. But starting with a few common genes and then kind of filling out more and more as per the question is probably the best approach. But the spectrometry thing is actually another angle to it, which is mostly to do with color. And so that's people are looking at wavelengths of colors reflected off feathers and things like that. And this is a different angle. So, the previous question that was asked either by you or somebody else, what is a subspecies? And I said, we need different kinds of data. So the spectrometry data would be another kind of data that goes in.
But that also is useful using case of identification for species and subspecies.
Yeah, you can say that, okay, the breast feather, the green plumage has a certain coloration pattern based on these spectrometry values. The other species has a slightly different coloration, and this is, how different that is. You can quantify it. That's what lets you do.
Okay. Thank you.
Yeah. Going, there's a question about molecular clock studies. Organisms which are extinct. So molecular clock is actually, you can say, a simplistic way to look at this is to understand how much mutation has happened and trying to understand how long it takes for that much mutation to happen. And we are only talking about neutral processes as of now. So when you have a clock-like pattern, that means that these changes accumulate steadily over time, and that is a clock-like pattern. But you can have, in a phylogenetic tree, you can have parts of the tree following a clock-like pattern and another part which doesn't follow that. So people test for these in when you're doing research with these sequences. So that's one way to kind of look at it. Can it be conducted? Yeah, because it doesn't really matter. I mean, different genes have existed, so if you can get the same gene for the extinct species and for the extant ones, that should not be the problem. Yeah.
Clock-like pattern. How is the pattern discovered on a biology degree in order to conduct studies?
Sorry, Govind,
The pattern.
can't hear you very clearly, unfortunately.
Govin, you can type in the chat box. Your voice is breaking.
Oh, how was it discovered? Oh, I don't know that. I don't know that. Yeah, I'll have to look that up. I can look that up and get back to you. Any other questions? Yeah, I think Govind has asked a few more questions about the molecular clock. Yeah, different parts of the tree could have different patterns because that may depend on what is happening with those species at specific locations or something like that. I mean, there are circumstances where things change rapidly, and for some it doesn't. So yeah, so different parts of the tree can have different patterns. That's correct.
If there are no further questions, then we can wrap this session up, I guess. Robin,
Sure,
think,
sure. Yeah, and I'm
is
happy
it-
to take more questions later. You can always write in, and we'll get back to you.
yeah. We can wait for a few minutes if you have any other questions. If not, then we will wrap this session up.
Sure. Yeah, thanks everybody for participating, and just please feel free to reach out through the portal, and Sonia and other TAs and all that will always kind of help us connect. And
Yeah, sure. Thank you, Robin.
yeah,
Thank you,
all right.
despite
Thank you
your
so much.
health issue, you are this
Yeah.
thing. Thank you,
Thanks.
everyone. Then we'll
Bye.
wrap this session. Then okay, bye.